Okay, let's get started. Clickers would be a good thing to pull out if you haven't done so already, which brings me to a couple of questions that I've had online discussion group and emails about clickers. I'm going to try and work to see where people are registered and everything else this week. So by the beginning of next week, we should be able to connect some of the clickers and names. I will just post those online. I don't have time to answer 150 questions about clickers. And if there are real technical problems with your clickers, the reason that you pay your $6 or $6.99, whatever it is, if you're using a used clicker, is so you can call them up or send them an email, customer service-wise. So if the clicker is malfunctioning, I have absolutely no clue about how to deal with that. That's why, again, you pay for them. So that being said, why don't we move forward and have our first clicker question for today. Anti-parallel in the Watson and Crick DNA structure means that the 5' and 3' ends of one strand are next to each other, 5' and 3' ends of the opposite strand are next to each other, base pairs are in the middle, structures a double helix, phosphates are uncharged. So let's give it a start. Again, feel free to discuss this. That Stedman guy. Ten, 5. I do this because people in other classes wanted me to warn them how much time you have left. So some of the comments up front is B looks way too easy to be right. And because Stedman always asks these crazy questions that the obvious one is never right. But that's not true. B is correct. So obviously I explained that properly. Or much more likely, Eric explained it properly when he was doing that. So our next clicker question is, relative to B-form DNA, a metaphase chromosome is compacted about blank times. And you can fill in the blank. I don't need to tell you numbers.
It's a factor of 10 different. And I think 10,000. Remember, because the regular is 1,000. One was 1,000, one was 10,000. B-form is more compacted.
Obviously, I don't remember because I thought it was.
I definitely don't remember these things. Or maybe it's 10 and 100. My mind is playing. It was almost towards the end of last lecture. It's 100.
Well, why would you wait for that?
It was towards the end of the lecture when you showed the two.
I left. Are you talking about Monday or Friday?
I think it was Monday.
Oh, then I didn't wait.
Maybe it was Friday.
I had to leave it on Friday.
There's too much information going in my brain. I can't tell where it came from. It's like I'm on a fire hose in my mouth. I'm just trying to drink.
10, 5. Okay, most people think C. There are basically two ways to answer this question. One is to remember what was on the slide, which is the easy way. And there's also the hard way, which is to remember how many base pairs per turn and how many base pairs you have in a human genome, general chromosome, and do those back calculations. That'd be a real pain in the neck. But you end up with the same number, which is 10,000. So that being said, I will let you on to a dirty little secret, and that is how professors who remain nameless love to write multiple choice questions. It's so much easier to have numbers as answers to multiple choice questions. So just again, a dirty little secret as far as that's concerned. So you may put away your clickers. One of the questions that somebody had online was, are there always going to be 2 clicker questions per lecture? The answer is probably there will be 2 per lecture. A makes it a little bit easier for me, but also if you do the calculation in terms of number of lectures where there will be clicker questions, you've got 25 of them times 2 gives you 50 possible clicker points. So also will give you sort of an idea where that is likely to be for the rest of the term. So today we're going to continue talking about why Watson and Crick's model was totally worthless, because that's not what DNA looks like inside the cell. It's really a whole combination of lots of different things. Start out with nucleosomes. This is sort of the first level of compaction. Hopefully it's not news to any of you that nucleosomes are what packages DNA. And pretty much all DNA is packaged in nucleosomes all the time, at least for eukaryotic DNA. We'll ignore bacterial and archaeal DNA for the next couple of lectures. Talk a little bit about chromatin remodeling because the DNA is so compacted all the time. Again, this 10,000-fold in metaphase chromosomes, but usually about 1,000-fold most of the time. You've got this big problem of how do you get that information, which is in the genetic material, to be read out. And so chromatin has to be compacted but also accessible at the same time. So remodeling turns out to be absolutely critical for that. Look a little bit at the 30-nanometer filament. We understand a lot less about this. Nucleosomes we got pretty well down. 30-nanometer filament much, much less so. And I'll talk a little bit about topology and supercoiling. This is a whole course, again, in and of itself. Unfortunately, I think our textbook does a really bad job of it, so I'll try and find a few more links online to talk about a little bit more about topology, and I'll spend a little bit more time on that. Then we'll talk about heterochromatin, hetero being different or not true versus true, euchromatin. Heterochromatin is really the main sort of compacted state of chromatin, and usually then means it's completely turned off. And that leads us to a little bit of a discussion of epigenetics. We'll talk a lot more about epigenetics towards the end of the course. But it's the state of the DNA. It's not the sequence of the DNA, which is leading to some kind of trait, which actually happens. How do you get heterochromatin? It's really about chromatin remodeling. A lot of that has to do with changes to histones, which of course gets back to nucleosomes in the first place. And they'll talk about how heterochromatin is controlled. And this is sort of the first step of that. Again, we'll talk a lot more about this towards the end of the class when we talk about gene regulation, because it turns out a lot of gene regulation has to do with that control of heterochromatin, or basically opening things up so that you can get proper gene expression to take place. And then, if we have time today, we'll talk a little bit about metaphase chromosomes right at the end. Just as a reminder, the DNA is not that nice orange line in the middle here all the time. Watson and Crick's wonderful model. It's bound to a whole bunch of proteins. So you've got not only all of the histone proteins, which are here, the nice orange circles with the crosses in the middle, but also a lot of non-histone proteins. And a lot of people forget, me included, that there are as many of these non-histone proteins around as there are of the histone proteins. But the reason that you see so many of these histone proteins is they're all the same, whereas all these non-histone proteins are really quite different relative to each other. So the cartoon looks great. That's, of course, not what it looks like inside the cell. Inside the cell, most of the time, it looks like what we have up here in this upper image. This is that so-called 30-nanometer filament. Again, surprise, surprise, about 30 nanometers across. But really, clearly, heavily complexed. Each of these sort of whitish balls here represents one of the nucleosomes. But clearly, they are really packaged very closely to each other. if you treat this 30 nanometer filament with various insults, you can end up with this beads on a string model down here. Probably still most of the time it doesn't look like this, but it makes it a lot easier to understand what's going on. And then each of these black blobs represents a nucleosome relative to everything else. Now, why nucleosomes? Well, nucleosomes relative to everything else are actually pretty small, and because of that, we understand the structure at an atomic resolution. So we know really, really well what the nucleosome structure looks like, and when the X-ray crystal structure of the nucleosome was published, actually almost 20 years ago now, it was by far and away the largest structure to have been solved by X-ray crystallography. Has all of the histones, 8 histones here in the middle, and then the DNA wrapped around the outside in slightly less than 2 complete turns. What's important about this slide is basically that the 2 ends, where it goes in and where it comes out, and this I'm referring here to the DNA, not the protein. These are the tails here hanging out as well, are really pretty close to each other, and it's completely symmetrical. It doesn't really matter which way these things are flopped relative to each other. And that means you can actually package the nucleosomes themselves together. How do you have interactions with these proteins and DNA? So kind of contrary to what we were talking about last time when we talked about the DNA structure, you got this wonderful major groove, and the alpha helix fits really well into the major groove, but that's really critical for specific protein interactions, protein DNA interactions. We're talking about nucleosomes. We want to have non-specific interactions. So most of the non-specific interactions that histones have with DNA in the nucleosome are not going to be in the major groove, but in the minor groove. And so if you look here, here's a bunch of amino acid side chains interacting in the minor groove. So there's a minor groove, here's a minor groove, etc. There's a major groove. Any idea what all of these red amino acid side chains might be? What's the charge on the outside of DNA? Negative. What do you want to interact with negative charges? Positive charges. What are going to be the amino acid side chains that have positive charges to them? These will be the basic side chains, which are lysine and arginine. So lots and lots of lysines and arginines, and we'll see that when we look at the sequences of these guys in just a second. So lots of charge interactions. There's also lots of hydrogen bonds which form between the nucleosome DNA component and protein component. As I mentioned before, you've got 8 different polypeptides, separate polypeptides, I should say, each of them present in 2 copies, and it makes it a little bit confusing if you're trying to write a question about this for an exam, but also to try and follow the whole process. And so I like this. Again, it's unfortunately not a figure from your textbook, but I like this assembly process because it gives you a better idea what's in fact going on, and again, I think gives you a better idea what the components are of the histone complex. So there are 4 different what's called core histones: H4, H3, H2A, and H2B. So these have different sequences. These are different primary sequences. I heard people talking about those a little bit earlier on. So that's the different sequence of amino acids. These guys come together, but they come together in 2 slightly different ways, which turns out to be really important for how you get chromatin remodeling and histone remodeling. H3 and H4 bind together and form a dimer. Now this is a heterodimer, not a homodimer, because they're 2 different polypeptide chains. And then you get a dimer that forms between these 2 dimers. So it's H3H4, which I've listed here, together, and a dimer of this dimer. So it ends up being a tetramer of these individual chains. So that's one part, and people always think about this, and I like to think about it as well, sort of the top of the nucleosome. That's where the 2 ends are coming in. Then you have H2A and H2B, but H2A and H2B dimerize with each other, but then associate independently with the nucleosome process. So instead of the tetramer that gets assembled, it's those two dimers that get popped in. And that becomes really important when we talk about alternative histones and, again, the nucleosome remodeling that we're going to talk about. To do this, molecular gymnastics, it's not terribly surprising. You have these chaperone molecules, so hopefully chaperones, so I guess it's the 1900s or 18th century, making sure that you don't have the wrong interactions. Like for my kids, chaperone, make sure you don't have the wrong interactions that happen. So it's basically helping have the correct interactions. And it turns out that most macromolecular assemblies, particularly when you've got complex, lots of different polypeptides that are coming together, polypeptides in DNA, very often you have these extra chaperones, which will make sure that you've got the correct interactions and not the incorrect interactions that are taking place. I mentioned briefly here the tails. These are these extra stretches of amino acids which are not inside the DNA when it's wrapped around inside the nucleome, also known as the tails. We'll see why that is in just a second. Why do you have tails? Well, these tails are first stabilizing the nucleosome, because it turns out they also have large numbers of lysine and arginine residues on their ends. So lots of negative charge that's going to, well, sorry, positive charge is going to interact with the negative charge on the DNA. But also, they're really important for linking nucleosomes together, because I mentioned before, it's merely that 30 nanometer filament where nucleosomes are really lined up right next to each other that you have most of the time. But also, these are now available to get changes to happen to them. If your protein's wrapped up on the inside of this whole loop of DNA, it's hard to have any kind of interaction with it. So those tails are really where the business end is of any kind of chromatin remodeling complex that takes place. So how do we know about nucleosomes? This is a real classic experiment shown here on the left-hand side of the slide. Basically, you make those beads on a string, as I showed you in the electron micrograph a little while ago, and then use very small amounts of a nuclease. Now, we haven't really talked that much about enzymes so far. ACE just means it's an enzyme, and I would say 99% of the time, what that ACE is doing is hydrolysis. So that hydrolysis, what does that do? Breaks things down. In this case, what does it break down? We'll tell you something at the beginning of that name. So it's a nuclease, so it breaks down nucleic acids. What it does in this case, if you don't use too much of it, it will just break down the nucleic acids that are not protected by the histones binding to each of the nucleosomes. So you end up with most of your nucleosome-bound DNA in these complexes with the protein and then this piece of DNA that's wrapped around one nucleosome. The key to this experiment is that you use very, very, very small amounts of that nuclease. So you end up with not just one of these nucleosomes together, but sometimes you've got two, sometimes you've got three. And then you can separate this DNA away from the proteins and then look at it on an agarose gel electrophoresis. And it's a little hard to tell here with this projector, but most of these pieces are down here, about 100 base pairs in length. But if you look here at the really short amount of times that you use this nuclease, about 2 minutes, now you start to see what people also call a ladder, but it's one nucleosome, two nucleosomes, three nucleosomes, four nucleosomes, and conveniently they've added it on the side here. Sorry, I don't think this was in the lecture notes I posted last night, but here's the link to it, and it'll be in the, the YouTube video when that ends up getting posted. This is also the way that you figure out what proteins are involved in this process, because you've chewed up all the DNA here. Then you look at all the proteins. So what are these proteins again? Let's look at those in a bit more detail. We mentioned before H2A, H2B, H3, and H4. Turns out that all four of these histones have a very similar structure to them in the middle of their primary amino acid sequences. And that's also known as the histone fold. It's just shown here as these 3 alpha helices because that's what it is. It's 3 alpha helices. And it doesn't matter if it's histone H3, histone H4, H2A, or H2B, they all have this histone fold to them, and that histone fold just loves to dimerize. So that's what we just talked about. We had the dimer of dimers, H3, H4, and then H2A and H2B, which are then coming together to form this structure here. Also want to mention, why do people call these tails? So these tails, which are not as part of that histone fold, people knew about way before they actually had the crystal structure of the nucleosome. And why is that? Well, because it's easy to get primary amino acid sequence. And these things are there which are not well conserved, not clearly part of that histone fold structure. So anything which is not part of the histone fold is a tail. You've got two kinds of tails. You've got N-terminal tails and C-terminal tails. And we'll see what's happening to each of those, although most of the business that goes on in terms of histone modification is in these N-terminal tails. Not exclusively, but most of what's going on there. So again, reminder, this is what that nucleosome complex looks like as a cartoon. We've got our 8 core histones in the middle here with tails hanging out at either end. Not surprisingly, because these ionic interactions, the hydrogen bonds, and some of the other van der Waals interactions, this is a pretty stable structure. But sometimes you need to change that stable structure. So how do you do that? Usually it's through these what's called chromatin remodeling complexes. Anytime molecular biologists talk about complexes, they mean that you've got multiple proteins together which are doing something. And almost all of molecular biology is due to complexes, lots of different things together. So here again is just a cartoon, and we'll look much more at these a little bit later on, of one of these chromatin remodeling complexes. Cartoon at the top. This is a higher resolution structure of what one of these things looked like down here at the bottom. Basically, it wraps around the nucleosome, and in most cases, it's actually an ATP-dependent process. You have hydrolysis of ATP, which you use to move the DNA along the nucleosome process. And that's supposed to be shown here by these little blue dots. So just sort of keep track of this one here going to here, this blue rotating over here, and that blue rotating over here. Now, why is this important? Well, it's probably because there's some particular sequence, specific DNA sequence in there that you want to interact with. You want to have another protein to interact with. So that's why you need to have these chromatin remodeling complexes. There are literally hundreds of chromatin remodeling complexes present in most eukaryotic cells. Yeah?
Is the chromatin remodeling complex the thing that recognizes the sequence on the DNA and decides that needs to be available?
Okay, so the question here, I'm just repeating it, is the chromatin remodeling complex recognizing a specific sequence on the DNA and then saying, this is why you need to remodel in this particular place? In some cases, yes. Usually, however, no. And we'll talk much more about that later when we talk about gene regulation, and a little bit here when we talk about heterochromatin as well. There's usually a different specific DNA binding protein that's involved that then will bring the chromatin remodeling complex to a specific place. Because, again, getting a little ahead of ourselves here, but nucleosomes are really common. You find them all over the place. And so those chromatin remodeling complexes are going to have to deal with, basically the same thing everywhere. And so it's more important to bring the chromatin remodeling complex to the DNA as opposed to have that chromatin remodeling complex per se just find that one particular place on the DNA. Yeah, sir? Question here. Yeah.
When you talk about generally in the hundreds range for eukaryotes, is that per chromosome or per cell?
So when I mention that, so again, the question, remind me to repeat questions, by the way, if I don't. When I say hundreds of chromatin remodeling complexes per eukaryote, what I mean is not per cell, but per genome. And so if you're looking at the genome, that genome complex. So, yeah, and the way that we can tell, in fact, if we look at the sequence, it's a lot easier to tell the sequences, look at the sequence and say, oh, that looks like it could be part of a chromatin remodeling complex. Again, a lot of this is protein families. So getting back to our discussion of protein families right at the beginning of class. Yeah?
Is amino terminal
Is amino terminal tail, amino terminal tail a different name for N-terminal tail? Yes, exactly. And as I mentioned, I think online, maybe it's just in an email, molecular biologists are lazy, and so they love to contract things. So yes, amino terminus and N-terminus are exactly the same. It's just a different way of saying it. Yeah?
Can you slide back? You said that you just use a small amount of nuclease, so it only breaks down the nucleosomes that are not protected?
Well, it only breaks down the DNA, which is not protected by Sorry, repeating the question. Said I used a small amount of nuclease, so it doesn't break down the DNA. What was it with nucleosomes? So I probably should have said it doesn't break down the DNA that is associated with nucleosomes. It only breaks down the DNA which is between the nucleosomes. I may have misspoken there. Okay, so I'll talk a little bit more about nucleosomes, and this gets back to your question about chromatin remodeling complexes and specificity, et cetera. One of the ways that we actually know a lot about how nucleosomes are assembled, and so you have the histones and the DNA assembling with each other, is getting homogeneous nucleosomes. As all of you should have said, well, X-ray crystallography. We'll do X-ray crystallography. The only way to do that is have something which is absolutely homogeneous. Well, how do you do that if these nucleosomes are binding to pretty much any different DNA? Turns out that nucleosomes like to bind to some DNAs better than they like to bind to other DNAs. And that has to do with the structure of DNA that we talked about last time. And that is that if you take a DNA and try and bend it, it's a lot easier to bend DNA that has AT base pairs where you make that bend. And so if you have a sequence of DNA which every 10 nucleotides, as we mentioned before, it takes 10 nucleotide base pairs to go around one turn, then those are going to be easier to bend. And so it'll be easier to wrap those around your nucleosome. So here, if you want to have a piece of DNA that binds to nucleosomes really well, say you want to do an X-ray crystal structure of it, then what you want to do is have a particular sequence of DNA that has AT base pairs every 10 nucleotides, and in fact, GC base pairs in between those. And so you can do that just specifically with this sequence. It turns out you can get what are called phased nucleosomes. So you can end up with a particular piece of DNA that has nucleosomes exactly where you want them to be. So if you want to study how proteins interact with nucleosome-bound DNA, you want to have one of these particular DNAs. So just binds a little bit better to any of these. Now, one of the things that we need to remember is, of course, all of this is happening inside a cell with a bunch of water, at least for us, at about 37 degrees Celsius. And what that means is, yes, even if you have a nucleosome complex, the DNA is wrapped around these proteins, but it's flexible. It's breathing all the time. And so if you do experiments, in fact, a lot like some of those nuclease experiments we talked about before, the nuclease that was used originally is something called micrococcal nuclease. It binds and will chop up, hydrolyze pretty much any DNA sequence. If you now have a specific nuclease, which just binds to and cuts one sequence, it will cut DNA which you put into one of these phased nucleosomes with a reasonable frequency. And what that means is that DNA is coming on and off the nucleosomes on a relatively slow, at least for biological case, but it still is coming on and off. And so here, 250 milliseconds, so about a quarter of a second, it's wrapped, and then for a very short period of time, it's unwrapped, et cetera. So this is that breathing process. And it turns out if you've got nucleosomes that have ATs every 10 nucleotides, they're not going to breathe very much. If you've got those that don't, they're going to be breathing a little bit more. And what that allows is some of these other binding proteins to even associate with DNA that is normally associated with nucleosomes. And so again, the way that you show that is by using one of these specific nucleases, which will then cut the DNA that's normally compacted with these nucleosomes. So that happens, but it's still not that common. And if you really want to have this happen, and particularly with all of the DNA at one particular place, then of course you need these chromatin remodeling complexes. So just as a reminder that we have these kinds of things which are going on there. So chromatin remodeling is basically moving the DNA along, but we also mentioned that these histones, when they're being assembled, have to have chaperones to help, make sure they don't do the wrong things, but really much more importantly, how they're doing the right things. Because you've got moving the DNA along, but in some cases, we'll see when we talk about the mitotic chromosomes, alternative histones, so that you've got to be able to move these things in and out from each other. So basically, here we already talked about the chromatin remodeling complex that moves the DNA back and forth around the nucleosome, but you also have histone chaperones, which again in an ATP-dependent process will mix and match the different histones which are there. Now, one of the things that you'll notice here is the exchange has happened of H2A and H2B. Well, you remember H2A and H2B are those that come in just as dimers. They're not the H3, H4 that's sort of binding at the top. That seems to be the most stable part of your nucleosome. So if you're exchanging the different histones, it's almost always going to be H2A and H2B. Although in some cases you can completely dissociate histones and then put a whole completely new histone core into these. This is a much rarer process, but all of them happen. But again, the key to this is really you need to use a lot of ATP hydrolysis to get all of these exchanges, and that's mostly because of the stability that you have due to all of these protein nucleic acid interactions that you have in the nucleosome. So this is great. We now completely understand nucleosomes, right? Yes, we know everything you need to know about nucleosomes, but that's not what the DNA looks like most of the time. It's usually in a 30 nanometer filament. So we need to add a couple of things. The most obvious one is histone H1. And histone H1 is the histone, which is also called the adapter histone, basically links the core histones, H2A, H2B, and H3 and H4, with the DNA together to build these 30 nanometer filaments. I put in here many kinds to remind me to mention that of the histones, H2A, H2B, H3, and H4 are incredibly similar in their amino acid sequence in all eukaryotes, from the mighty yeast to the mini human. These sequences are practically identical to each other, amino acid sequence-wise. And what that tells you is that this is something which is clearly very important and has been conserved through certainly all eukaryotic life, and it turns out there's some histone-like proteins in archaea too that have some sequence similarities here. So these things have probably been around for a really, really long time. On the other hand, histone H1 is nowhere near as conserved. There are lots of different sequences that you have in histone H1s. So H2A, H2B from humans and yeast is, I think it's something like 89% identical at the amino acid level, which means what in terms of its structure? Yes, the structure is absolutely identical. There's no change whatsoever in the structure. Again, not terribly surprising because these things clearly have to do very similar things to each other. What does H1 interact with? Interacts with the tails of those core histones, either N or amino terminus or C or carboxy terminus, and is really critical for putting together this 30 nanometer filament, but together with all of these other proteins. This is just, again, to remind me that there are lots of other proteins. Some of them they're shown here as being sequence-specific DNA binding proteins, but also you have a lot of non-specific DNA binding proteins that are involved in this process. And so basically it's going from this beads on a string, each is individual nucleosomes, and getting them packaged into something that looks a little bit more like the DNA that you purify from a cell. And when I say a little bit more like, because we don't actually really know what exactly that looks like, and it's partly because you don't have a nice homogeneous population of exactly the same thing, so you can't do X-ray crystallography. All the other structural techniques don't really let you get very close to looking at this. So everything we'll be showing in the next couple of slides are just models for how people think these things fit together. These are models which fit the data, but there are a number of different models which will be fitting that data. And so up here, shown what people think these two nucleosomes are going to be looking like together with each other, and then you expand these nucleosomes. You remember I said that the nucleosomes are pretty much like disks, and they're very similar. Doesn't matter which way round they are, and they can also pack on top of each other. So certainly that nucleosome packing seems to be pretty important. Yeah? So the question is, during replication, are nucleosomes being made at the same time as DNA? The answer is yes, and we'll get back to it when we talk about replication in terms of moving these around. Because you clearly obviously have a problem. You have one copy of DNA with all the nucleosomes that are associated with it, and then you're making another copy of the DNA, which you also then have to package with the nucleosome. So it turns out, and I didn't mention this before, but we'll talk about it again later, that the histones are among the most common proteins that you find in any cell, because you need a whole bunch of them to compact your DNA. So, and also they're being expressed at very high levels. So you've got a lot of them. Yeah, so we'll...
Most abundant protein, is that what you just said?
Among the most abundant proteins. So the histones are among the most abundant proteins that you have in the cell, in eukaryotic cells. So again, more models of what this 30 nanometer filament looks like. EMs, which are fuzzy on my screen and probably even more fuzzy up here. Again, these are all ideas that people have on how these 30 nanometer filaments might fit together. Now, why are we spending so much time making all these models? Because it's what most of the active DNA in the cell looks like, is this 30 nanometer filament kind of structure. The last one that I wanted to show here is this structure. And again, it's all about these disc-like nucleosomes packing together. And this is really sort of an alternative model to that zigzag model, which I showed a couple of slides ago. This is now much more of a helical model. And this structure actually has been determined. So we know that that is the particular structure. But again, this is a completely artificial environment which was put together to make this structure. So it may or may not be that the 30 nanometer filament looks like this. And it's quite possible that multiple models are correct. So there are lots of different ways to theoretically skin the cat, as it were. So let's look at these H1s a little bit more detail. This is one specific one of those Histone H1s. Now what you can see is your nucleosome, which would normally stop here, has an extra piece, and there's your Histone H1, which is attached to it. If you do one of these micrococcal nuclease experiments that we talked about before, we see a ladder of 150, excuse me, 300 nucleotides, 450, 600, etc. Here, you'll end up with 170, 340 nucleotides, etc. So it protects about 20 more nucleotides from digestion by micrococcal nuclease. In these 30 nanometer filaments, the tails of the histone, core histones, are clearly very important for interactions with the other histones in these 30 nanometer filaments. Almost none of the other structures and those models actually have where these tails are interacting. And so if you look in pretty much all of these models, you'll notice that they're really skinny and floppy. Well, that's because we don't know where they are. Because in those crystal structures, all the structures that we have so far, these particular pieces of the histone sequences are not structured. So we don't know what these structures actually look like, and so we can make all kinds of cute models like we have down here to take a look at that. So come on, to continue on the H1's theme, we do now have some indications, at least again in some other structures. This is now not using X-ray crystallography, but this is a new technique called cryo-electron microscopy, which there's a new professor in chemistry, Steve Reichow. I don't know if any of you have heard about this, but he's now starting up his lab down at the CLSB on the South Waterfront, using some of these new microscopy techniques to try and analyze single molecules. So instead of having a crystal, we've got to have all actually identical. If you look at these single molecules, hopefully we'll be able to get a way to get an idea of some of these structures without having to have so many completely homogeneous molecules. And so that's just an example right here of one of these nucleosomes, together with Histone H1, now with electron microscopy, and you can put this together again to try and form sort of a 30 nanometer filament. And that reminds me, there's a really great website that's linked in the DNA links in D2L, where they have videos, well, they're all animations, of course, of how people think these nucleosomes are interacting with each other, and then how they form these larger filaments, and then eventually getting to the metaphase chromosomes. Yeah? Okay, send me an email, remind me. I can also just put some links. They're on YouTube as well. Sorry, the question there is you can't get them to work. And so, but just please remind me, and I'll try and make sure. If there are any links on D2L that are not working, please email me, and I'll try and figure out what the problem with those are. Okay, now we start to get to bigger and bigger pieces of DNA, which are still, nucleosomes, we got down. 30 nanometer filament, we got some ideas. Beyond that, we're getting on thinner and thinner ice. It's really a lot more speculation on what's going on there. So first of those is what happens when we've gone from 30 nanometer filaments to much larger ones, and most of those are about on the order of 300 nanometers. We're still trying to make that 10,000 that we had right at the beginning here. These are usually what are called loop domains, and we know this because when we look at a couple of particularly amphibian chromosomes, you can see these specific looped. But the important thing here is it's a domain. Now, unfortunately, the molecular biologists don't have that big a vocabulary, so they talk about protein domains. It's a separate, folded piece of protein that's a particular function. When you talk about a domain of a chromosome, that's a piece of the chromosome that doesn't necessarily have a function, but it's usually kind of separate from the rest of the chromosome. And so usually each of these loop domains has one particular activity to it. We know there are about 2,000 of them in human chromosomes, and holding them together are a lot of those other non-histone proteins that you find when you look at DNA. And so these are the scaffolding processes, and then they also include these so-called matrix attachment regions. So let's look at cartoon of what these things are supposed to look like. We've got 30 nanometer fibers, and each of these loops here would represent, well, again, what people will call a domain. And it turns out that they're usually co-regulated. So all of the genes that are in this particular loop are going to be regulated in the same way. But to hold all these loops together, you have this scaffold down here at the bottom. And that scaffold is binding to specific sequences, some non-specific sequences, and also other sequences that are present inside the nucleus. And so it's really providing this structure. And so I don't have a picture of that now, but when we talked about the 10,000 compaction, you also had the 30 nanometer filaments bound to this sort of glob in the middle in the micrograph. That glob is all those scaffolding proteins. So everything is being held together there. So as an overview, what have we talked about? Again, Watson and Crick. It's not what the DNA looks like, but it's a nice model. Beads on a string is also not what DNA looks like, but we know a lot about each of these individual nucleosomes and how they're interacting with the DNA. Oh, excuse me, over here. Keep forgetting there are two screens. And then these nucleosomes get packed together into these 30 nanometer filaments. We're not quite sure how that works. And again, your understanding is going from big to small here. The extended section of the chromosome, these are these looped domains, which again have lots of scaffolding proteins down here at the bottom, which will be interacting with them. These loop domains get wrapped up together, and we'll talk a little bit about that probably towards the end of class today. And then finally, each of these loop domains gets compacted into your metaphase chromosome. So from 2 nanometers to 1,400 nanometers across, but the compaction in length is much, much, much higher than that. Again, up to 10,000 here. So I mentioned supercoiling. This is another way that you can compact a helical structure. And these 30 nanometer filament domains are all highly supercoiled. So what do I mean by supercoiling? Now, one of the really neat things about Watson and Crick's structure was it's a helical structure. Things are wrapped around each other. Well, whenever you have a helix, it can be wrapped around each other, like we have this helix, but also that helix can wrap around itself as well. And so that's what supercoiling is. So it's a helix wrapping around a helix. So something is coiled already that gets wrapped again. Here's the example of the cartoon up here. We have a non-supercoiled DNA, and now here, what most people think about as a supercoiled DNA. We have the strands that are wrapped around each other. And so I'll just do that here quickly with my ancient technology here. So there's supercoiled. So we had a coil, it's now supercoiled, so it's coiled around itself. With DNA, since you have two strands that are wrapped around each other, you have two basic kinds of coiling which are going on. You've got the coiling that takes place with the two strands around each other, which is what people call twist. And then there's where the two strands are crossing each other. Now, unfortunately, this is just a single strand because of the phone cord. This is where you've got those double strands crossing each other. That's called writhe. And the supercoiling is best defined by something called the linking number, which I think we have down here at the bottom. Yeah, so L at the bottom of this slide. Sorry, maybe a little bit hard to read here. But L is just a combination of the twist and the rise. The twist against the two strands wrapping around each other, and the rise, those strands crossing across each other. How many base pairs do you have in one turn of B-form DNA? 10 base pairs per turn in B-form DNA. So if you have a 360 base pair piece of DNA, like we have right here, how many twists do you have? How many times do you go around? 360 divided by 10 base pairs per turn. How many turns do you have? 36. Exactly. So that's what we have here. And this is what the T stands for. It's a twist of 36. So 360 base pairs just divided by 10. There's also what's called a relaxed DNA here. Now, if you take this DNA and put some extra twists into it, and then allow those to interact with each other, now we end up with these strands wrapping around each other. Now, it turns out that twist and writhe, which are these, double strands crossing each other, just the single strands wrapping around each other, these are completely interchangeable. So you have writhe or twist, and all that depends on now is the change in the wrapping around with twisting. So if you untwist, you're going to be taking out some of those changes, or if you wrap them up together. So you may have noticed I've been fiddling around with the ends here. Basically what I've done is I've taken my, now again it's a single helix but not a double helix, but DNA works exactly the same way. If I take this now and I put some extra twist in it, so I'm wrapping it around itself, this is equivalent to getting your writhe. So this wrapping, it's twisting is equivalent to wrapping. So basically what that says here is that this guy in the middle, which again, what most people are talking about supercoiling, where you've got the double strands crossing each other, is exactly equivalent to this structure over here, where you've untwisted the two strands. They're completely interchangeable relative to each other. Now, so why do we care? Yeah, who cares about this? Well, it's because all of the DNA in the cell is not like this, because this would take way too much space. It's all like this. But both writhing, so double strands crossing each other, which gives you nice compaction. And again, you think about this. This is one way to have it, and then we wrap it up. This is, clearly a more compacted structure. So I don't want to lose the ends, because if you do, it's a problem. So that's more compacted than it would be if it's not. Let's put the two ends back together. And that's exactly what you have to do. You have to do this to compact your DNA. But at the same time, the negative supercoiling, I haven't talked about positive and negative yet, that's a change in that linking number that I talked about. So here in the middle, we have a linking number of 32 because we have changing this by 4. It's a lot easier to think about here. We've now taken 40 base pairs and pulled them apart relative to each other. We've untwisted them by 40 bases. In order to get this into the most stable state, it's going to be here in the middle where it's wrapped back around. But that negative supercoiling, and negative because it's taking your linking number and lowering it, is again equivalent to untwisting your 2 DNA strands. Why do you want to untwist DNA? So yeah, you pull them apart, but then also a comment up here, you can replicate it. In some cases, you really do need to have those base pairs which are available. So it turns out it's absolutely critical for replication and absolutely critical for transcription, making that RNA copy from the DNA. So you have to have that hydrogen bond information. So negative supercoiling is awesome because it allows you to have compaction and have the 2 strands apart. So it's an incredibly useful thing, which of course is why it's probably evolved to have this particular process. It also turns out that there's a few cases where you have positive supercoiling, and Travis is not allowed to answer this question. Where do you think you would find areas where you want to have your DNA overwound so it doesn't come apart as easily? Okay, so the one comment up here in front was you want to have them in coding genes. It's probably not so good because you want to have where you're going to be able to actually access the DNA. So I'll give you a hint, also because we're getting toward the end of class today. But if you're at very high temperatures, we talked about DNA coming apart, and that's going to depend on the GC concentration. But if you're supercoiled, if you're negatively supercoiled, they're going to come out a lot easier. If they're positive supercoiled, they're going to come out a lot less. So you've got a very high GC organism. You might want to have positive supercoiling. Or in the case that, again, Travis is not supposed to answer, is if you're working with organisms that grow at extremely high temperatures. So you have an organism that grows at 80 degrees Celsius, like ours, which is usually way beyond the T end of most DNA, or 90+ degrees Celsius, which Dr. Bartlett, some of you have had him for Principles of Biology, he was an organism that grows at 90+ degrees Celsius. One of the few ways to keep your DNAs together is to have it be positively supercoiled. So it's actually a lot harder to pull the 2 strands apart. So that's what we have down here, supercoiling mostly negative. And this linking number, as I mentioned before, it's just the twist, which is the 2 strands wrapping around each other, plus the rive. If you don't cut your DNA, then that linking number stays constant. So these 2, as I mentioned before, are really equivalent to each other. How do you increase or decrease supercoiling? There are these things called topoisomerases. We'll talk a lot more about them later on. Topoisomerase 1 changes your twist because it cuts just 1 strand, and topoisomerase 2 changes rive and cuts 2 strands. And again, we'll see why you need these proteins a lot more later on. But again, it has to do with why negative supercoiling is such a good thing. If you want to have negative supercoils, then somehow you've got to introduce negative supercoils into your DNA after replication, for instance. So topoisomerases, it turns out, are absolutely critical for DNA replication, condensation of DNA. They're also the targets of a number of chemotherapy drugs because you're talking about replication. You have to have supercoiling going on. Also great antibiotics because it turns out that the bacteria have exactly the same problem. They also have to compact their DNA. They also use these proteins to do that. So there are drugs which are involved in this. Last thing I wanted to talk about in terms of supercoiling is that wrapping your DNA around a nucleosome also generates supercoiling. So any kind of compaction that you're getting with the nucleosomes also gives you, conveniently enough, negative supercoiling. So again, helps those 2 strands to come apart. This is a cartoon of how that works. Again, you associate nucleosomes with your otherwise normal, just twisted up DNA. As soon as you put in nucleosomes, because your linking number doesn't change. So here they're bound to nucleosomes at one end. To compensate for that, you have to have rise going on at the opposite end, where you wrap your two strands apart from each other. And then over here on the right-hand side, if you put in a topoisomerase, which is now going to change your linking number, and then you take away all of your nucleosomes, you end up with this different sized pieces of DNA. And again, we talked about how are you getting compaction. Now, why do you need the supercoiling? It's to compact your DNA. Well, nucleosomes compact the DNA, but supercoiling compacts it as well. If you look at an agarose gel electrophoresis, and we'll talk more about gels and things at the end of the class, but basically the smaller DNAs are going to go faster than at the bottom of your gel. The larger DNAs are going to go slower. So the more and more nucleosomes you have associated with your DNA, put in topoisomerase, reattach the ends, you end up with more and more of this very compacted DNA. So nucleosomes are helping out there as well. Questions about supercoiling? And again, I'm more than happy to talk about these on D2L as well. Because again, unfortunately, the textbook really doesn't get into this at all, but I think it's absolutely critical for how molecular biology works. Okay, but again, I'd say I'm more than happy to talk about this in office hours and play around with my helices and supercoiling and so on and so forth. Okay, let's move on and talk about heterochromatin. Last 10 minutes or so of class. This is an example of epigenetics. We'll talk more about some of the other epigenetic mechanisms again later on in the course. But the whole idea of an epigenetic process is that it's an inherited process, something which is inherited, which is not a change in your DNA sequence. It's not a change in the genetic sequence. So epi outside of a genetic change. And so this is just an example here of your genetic inheritance. Usually has to do with a gene which is turned on or a gene which is turned off. Here we have gene X. You have a sequence change. This is now a genetic change which turns off that gene. That's now maintained as you have multiplication of each of those individual cells. On the other hand, an epigenetic modification, we have gene Y on. Here we're going to talk about heterochromatin. That heterochromatin is going to shut down that DNA and turn off the gene. That can be inherited. And again, we'll talk about how that epigenetic inheritance takes place for heterochromatin. But you can also reverse this. And this usually happens in the production of germ cells in terms of sexually reproducing eukaryotes. So this epigenetic change is a completely reversible one. Genetic changes, you've got to change genetic material. So the reversibility is actually a lot harder. Let's look at some of these heterochromatin examples. Probably the best known heterochromatin example are bar bodies, which again are unfortunately a little hard to see in this image here. Fortunately, we've got our arrows to show you where they are. If you look at nuclei of female cells, they've got 2 X chromosomes, one of which is inactivated. That inactivation process has to do with compacting that particular structure. So also known as the bar body. It's a, as far as we can tell, random process in terms of which X chromosome gets inactivated during development. And when you undergo meiosis, each of the cells divide. Oops, I just completely disconnected things here. That's interesting. When you undergo meiosis, that process, see if this will come back, is reversed because those individuals, get this to work. Yes, there we go. You have to be able to separate those 2 chromosomes relative to each other. And that whole compaction that takes place, it's the whole chromosome. It's also compacted whatever space you have to have the cytokinesis which takes place to pull those 2 chromosomes apart from each other. So this is the heterochromatin which always see again in female cells. So what's called facultative, and why it's called facultative is because it's reversible. Again, anytime you're undergoing meiosis, you've got to be able to unpackage that, repackage it, go back and forth. There are some cases which are so-called constitutive heterochromatin, and that's where the DNA is compacted all the time. Those are what you have that takes place at centromeres and telomeres. Hopefully centromeres and telomeres mean things to people. Yes? Middle of the chromosome, end of the chromosome, middle of the chromosome, which you use to pull it apart with. About 10% of any chromosome that you look at is going to be in heterochromatin. Most of that's going to be centromeres and telomeres. Yeah, question?
The book on this section really made it sound like most of it was just in that inactivated state. It didn't really talk for it. From my recollection about this facultative element of it, so it can go back and forth from hetero to euchromatin?
Right. So the question down here is that most of the, again, I'm paraphrasing here, correct me if I'm wrong, that most of the heterochromatin is facultative heterochromatin. So it is compacted some of the time, but it doesn't have to be compacted all of the time. And so it's that constitutive heterochromatin which is compacted all the time. But it's a relatively small amount. Okay. Okay, how do we know about heterochromatin? Well, it turns out that this is the awesome power of genetics, which they bring up again and again in our textbook, but it's a really neat example. Basically, in a normal cell or a normal fruit fly, you have heterochromatin in one place and euchromatin right next to it. And this is best known for the cases of the constitutive heterochromatin, things like telomeres or centromeres. And what happens here, and just look at the fruit fly, is in some cases what you get are genes that are normally in euchromatin, are normally expressed. So in this case, the white gene, and these damn geneticists, they always name things based on their mutant phenotype. So if you make a mutation in the white gene, then your eye is white. If you don't have a mutation in the white gene, the gene is red. Or the eye is red, I should say. So white's referring to the mutant phenotype. So here, normal fruit flies up at the top, white gene, which it turns out is actually relatively close to some heterochromatin, because you can figure out where it is relative to the end of the genome. You have normal red-eyed flies. On the other hand, if you have an inversion event that takes place during development, what you can end up with are these very bizarre-looking flies. They've got partially mutant eyes and partially non-mutant eyes. Of course, the mutant is now white. And so this is what's called a position effect. It just depends on where the gene is in the genome. It's not a genetic, well, it's a genetic change because you changed where it is in the genome. But it's no change in the gene itself. And this really confused people for a long time. Because they said, hey, what the heck's going on here? Our gene's normal. The sequence is normal. But we have all these changes in how it's actually being expressed. So eventually what they found was that it was a change in the chromosome. And the main thing about that was it basically removed this barrier between the heterochromatin and the euchromatin. How that happens, we'll talk about on Friday.